Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Genetics. Genetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate datasets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Austin Argentieri, who's a research fellow at Harvard and the Broad Institute and works in the Analytical and Translational Genetics Unit at Massachusetts General Hospital. He completed his PhD and postdoc at the Big Data Institute at the University of Oxford, and his research today is focused on large-scale analyses to understand the genetic, biological, and environmental determinants of human aging and aging-related diseases. So, Austin, without further ado, thank you so much for joining us today. I really appreciate it.
Thank you for having me. It's my pleasure to be here.
I'd like to jump in with a completely unfair and high-level question, but hopefully you can help us navigate this, which is really about aging. You study aging, but what is it exactly? We all experience this. We live it every day, but it's one of these things that's surprisingly difficult to put your finger on. Is it the absence of disease? Is it an entity in and of itself that's happening on a cellular or molecular level? Maybe you can enlighten us around what aging is and how we should think about it.
It's a tough question because I think if you look at the field, there's not great consensus among researchers in the field itself, what the correct definition of aging is. There's a lot of ongoing efforts right now to try to come to that consensus. But if you look at 10 different papers on aging, you might find 10 different definitions of what aging is. But I can give you a couple different perspectives on ways that I like to think about it. We can think about it from a population level in terms of life expectancy or in terms of this phenomenon of increasing mortality risk over time within a population. We can think of it on a sort of cellular or biological level in terms of some people like to think of it in this kind of like accumulation of damage kind of framework where you have these accumulation of deficits and sort of biological genomic damage and instability that happens over time, which leads to sort of a less efficient functioning of biological systems, which leads to increased risk for mortality, morbidity, frailty, dysfunction, all these kinds of things. There are a number of kind of cellular hallmarks of aging, a number of which have been studied so far. There are about 12 or so that we agree on might be key aspects of the aging process on a biological level, everything from shortening telomeres to genomic instability, loss of protein stability, gut dysbiosis, mitochondrial dysfunction, things like that. But on a sort of higher level, outside of the weeds of the biology of what's going on, it's really this phenomenon that across time, we have this increasing tendency towards failure, frailty, dysfunction, and increasing mortality risk. And we just kind of see that going up and up and up with sort of chronological age in the march of time.
Amazing. And you published a landmark paper, I think, pretty recently that is a culmination of probably at least a year or two of hands-on work, but also likely many years before that of thinking and refining your thinking about this problem and how to tackle it. Maybe you could talk a little bit about that. What was the original question you sought to try to answer? Like, what was the kind of beginning of this? And also, where did you end up? What did you find? Sometimes the beginning and the end are very different in these kinds of...
Sure. If I can, I'm just going to start sort of from the broader rationale for why we wanted to do this work, and then I think that will lead into what the actual work was. But my particular interest in focusing on aging comes not from necessarily the idea that I want to discover some therapy that's going to make us live to 120, 130 plus years. But I think studying aging and studying aging biology is a really, really effective and strategic way to try to address some really crucial public health problems, mostly premature mortality, rising rates of global multimorbidity, chronic disease, major causes of death as a public health problem. And the reason why aging is a really strategic way to do that is because we have these known sort of biological hallmarks of aging, some of which I mentioned before, that are known through, animal studies, but also human studies as well. But the key insight is that most of these hallmarks are shared across most major chronic diseases. And we care about these chronic diseases because a lot of them are the major causes of death worldwide. So the opportunity in thinking about aging is really that you have this kind of shared aging biology that's common across a lot of major chronic diseases that also happen to be the major causes of death in the global population. So the real rationale for thinking about the project that we've been doing recently is, well, if we could take advantage of that somehow, if we can start to be able to quantify signatures of aging on a biological level, it will help us to better come up with better sort of preventative tools, predictive tools that might be just one tool for many different chronic diseases combined, but will also tell us something about the common biology, the common genetics across all these diseases, which opens the door for therapeutic development and all these kinds of things. There's been a lot of previous research trying to come up with biological signatures of aging, mostly through things like epigenetic clocks or DNA methylation age clocks. We can get into, the pros and cons of the pitfalls and the strengths of trying to measure aging as a biological phenomenon from epigenetic information. But our specific work on this, which we just published recently in an article in Nature Medicine, is really focused on instead using large-scale population proteomics data to create accurate estimates of biological age across diverse populations, and use that then as a signal that we can use to predict mortality over 10 to 15 years in the future, many major chronic diseases and cancers, as well as lots of different aging phenotypes, frailty, biological decline, all these kinds of things. So this work that we've been doing most recently, it was in the context of a population cohort study called the UK Biobank. And about a year ago, they published this incredible resource, which is they released large-scale proteomics data for about 50,000 participants in the UK Biobank. So we had this sort of unprecedented opportunity to do really large-scale proteomics to try to capture aging signals or a signal's biological age. To do that, we took about 3,000 proteins from blood plasma, and we used those in the context of the UK Biobank. We used different machine learning models to take those 3,000 plasma proteins to estimate somebody's chronological age. Turns out you can do that with about 94, 95% accuracy, which is fairly high. And the interesting thing is then you'll have two phenotypes for a person. You'll have their calendar age or their chronological age on one hand, and then you'll have their protein predicted age on the other. Since the accuracy is very high for the proteins predicting age, for most people, those two values are very similar. But the people who are really interesting are where those values start to diverge a lot. So you'll have some people where they might be 50 years old, but their proteins predicted their age 40 or their age 60. So a lot of people fall within this 10 years plus or minus from their calendar age. And that phenotype there, which is really the difference between what your proteins predict your age should be and what your calendar age actually is, turns out is very highly predictive for mortality, many different chronic diseases, and many different age-related outcomes. So we took that phenotype, we call it a proteomic age gap. Some people in the field also call it accelerated or decelerated aging. But again, it's just this difference or this comparison between your sort of biologically predicted age and your calendar age.
Very different branding on those two, isn't it?
Well, historically, the epigenetics community has liked to just call it acceleration. I think there's a lot of pushback against that recently, thinking that that's probably not the correct terminology because that implies sort of a change in rate over time. But a lot of these clocks are calculated from some kind of cross-sectional snapshot. So I think people have been moving more towards this age gap concept, but whatever you want to call it, the concept is still the same. We're relating your biological predicted age to your calendar age. We then tested that in relation to sort of future disease risk for all-cause mortality. All of the major causes of death in the UK Biobank and a number of different cancers found out that this one single sort of biological estimate of your proteomic age is highly predictive of all the major non-cancer causes of death in the UK Biobank, 4 different cancers, but even with 50,000 people or about 45,000 people, there's not many incident cases of cancer over time, so we were a little less powered to study there. But also was related to all kinds of functional decline that you would expect with aging, so things like a comprehensive frailty index, walking speed, grip strength, but biological signatures of aging as well, like telomere length, IGF-1, liver kidney function, things that are very well studied in the context of aging research. But we also found that this signature of proteomic aging is still associated with all of these age-related sort of frailty, functional status phenotypes, even in those who have no lifetime diagnoses of any major chronic disease, which tells us that we're really picking up some signal of aging biology that's happening, in the years or decades before disease happens itself. So it's really this kind of like preclinical status, which makes a lot of sense. I mean, from the day that you have a diagnosis, it's not like the disease process on a biological level started that day. It's really been brewing up in the years or decades before then. And so I think what we show in this paper is that you're able to capture those developments far in advance of the actual disease diagnosis itself, which makes it exciting from a sort of preventative, sort of early detection point of view. And, I think what we ended up showing in this paper is that, aging, I think if you can sort of begin to quantify these aging estimates and phenotypes in the right way, they do kind of make good on this promise that you can find a common biology across a lot of these major killers and causes of death, and just from one estimate, use that to predict future risk of many different diseases. One of the things that we did in this paper that I think is really lacking in the field in general is it was really important for us to then validate the signature in other populations that are very sort of genetically, geographically, and sort of more profile-wise distinct from the cohort we trained these models in. So we developed this model in the UK Biobank. It's about 45,000 people. They're mostly white Europeans, very few diversity in that cohort, and especially in the subset of proteomics participants. So then we validated these models in the China Kadoorie Biobank. That was about 4,000 people, all in China. Very distinct genetic population, but also a very different cohort design. It's kind of the proteomic subsample. It's an ischemic heart disease case cohort design, which means they took half of the population for proteomics, their ischemic heart disease cases, and the rest is a random subsample of the cohort to mimic those larger cohort dynamics. So very different genetic ancestry, very different morbidity profile. We also validated the model in the FinGen Biobank, which is also a very distinct genetic ancestry group within the sort of European continent, but also all of the participants that they did their proteomic profile on were healthy and had no mortality during follow-up so far. So again, a very different morbidity profile, very different genetic ancestry, very different geographic population, and also a very different age group. The thing I didn't mention is when we train these models in the UK Biobank, we train them on participants between a range of 40 to 70 years old. But it was important to us to test, do these models still work well to predict somebody's age outside of those parameters? It was a pretty small window, but since the data are so large in the UK Biobank, it was the best place to try to train these. FinGen, on the other hand, they have participants from 20 to 80 years old with proteomics data. And what we show is that this model is fairly robust across both of those very different populations, basically the same accuracy for predicting age from blood proteins if we take the model and use it to validate in these two other populations. So I think that was a step in the right direction for the field. You don't see a lot of replication and validation of this kind of aging research across diverse populations. To be frank, you don't tend to see a lot of large-scale human population studies in aging right now compared to the just absolute ton of animal studies at the moment as well.
Yes.
So one of the things that we're hoping to, in our group, sort of push people towards and hopefully kind of lead the charge in the direction of let's make use of the wealth of incredible large-scale, human biological genetic sort of omics population data sets that are available to try to study and get to the bottom of some of these questions. But one interesting thing we learned is that not all model types are equal for this. We tested a lot of kinds of models to estimate biological age and then use it to validate across these different populations. And some of the really popular ones that everyone loves don't work very well for this. So for all the neural network enthusiasts out there, I'm sorry to say they don't work very well in making generalizations from biological data, at least from what we see, beyond the very specific parameters on which they're trained. So like I said, the UK Biobank trained in 40 to 70 years old. And if you look at the graph from a neural network making predictions, in FinGen, for example, you just see this sharp cutoff at 40, and it can't learn how to make predictions of someone's age below that. So That was sort of an interesting side observation for us, is that I think there's a lot of work that goes into statistical modeling to try to think about what's the best accuracy we can get out of this data set. But I see far less attention being paid to, well, what's the ones that give us the best accuracy in this data set, but the best generalizability across very diverse and different populations. So that was a good lesson for us as well. And we were happy that, it ended up we could use a much less expensive and complicated model type that was far more generalizable.
Yeah.
So
what ended up working best for the machine learning nerds out there who want to know?
We use a gradient boosting model called LightGBM, which is very similar to XGBoost, but it uses just a little bit faster. But it's in this family of sort of tree-based models similar to random forest as well.
Yeah, makes sense. I have a million questions on this, but first I want to say it's amazing and fascinating work. And I just to play it back to make sure I'm summarizing it correctly. So you're able to use this 3,000 protein score, combination of weights across the 3,000 proteins to predict a biological age, for lack of a better word, that's predicted by the proteomics, and compare that to their actual calendar age. And it's the residual that's interesting. People who are 10 years older, according to the proteomic score, compared to what the calendar says, are at increased risk for all the major non-cancers and many of the cancers. And it sounds like there was a power issue here, not necessarily that it's not predictive of cancers in general, but you just don't necessarily have enough data to say that definitively. Is that a rough approximation? Have I got that right?
Yeah, I mean, there might be some cancers where it's not involved at all. Some cancer signals we see were very null, and I don't think that would change, if we had a lot more cases all of a sudden. But for some of them, it certainly was the case that we only had, fewer than 100 cases, so it was difficult. Yeah. Forgot to mention is that one of the interesting things we learned is we start with 3,000 proteins, but it turns out you only need far fewer than 3,000. So
that's exactly where I was going to go. They're not all created equal, right? What did you learn about the proteins that are really driving this difference?
So it turns out whether you use, we identified about 204 proteins that are important for predicting age. Turns out if you use those 204 versus all 3,000, there's virtually no difference in the accuracy of your model. So what that tells us is you don't need thousands of proteins to capture sort of the complexity of what's happening in terms of aging biology. But turns out we sort of took that further and we went all the way down to 20 proteins and found out that you can still capture about 91% of that full model's complexity with just 20 proteins.
Wow.
And if you make a score based on those 20 proteins, it's associated with all of the same sort of multitude of aging phenotypes and incident diseases and mortality as the 200 protein score. So we're excited about the 20 protein score because you're reducing the complexity a lot in terms of information that has to go into your model. But you seem to be still getting all of the sort of high dimensional associations out of that. And the 20 proteins are very diverse that go into it. They span across extracellular matrix, inflammation, metabolism, some cancer stuff. So even though it's 20 very different proteins. Some people in the aging field like to focus just on inflammation or just on other things. We see some inflammation in there, no doubt. But one of the takeaways is even when you go down to just 20 proteins, it's a very multidimensional set of 20.
Yeah. Now, is it those proteins themselves, just thinking about the genetics of this, is it those proteins themselves that there are 20, genes, for example, that turn out to be major drivers of accelerated or decelerated aging or aging gaps? Or is it that those are, somehow kind of hallmarks of much more complex processes and you're measuring these proteins as outputs, but actually the genetic or environmental differences are happening all across the genome, but for some reason those 20 proteins are kind of capturing all the signal. What is it about those 20 proteins that makes them so predictive?
Yeah, it's a good question. I couldn't tell you that I have the definitive full answer to that question, but the interesting thing I think about proteins is that they're kind of this accumulation of genetic influence, plus they're also really good biosensors of environmental influence. So whatever you're getting in terms of the protein levels are ultimately going to be this combination of those things. There's been a lot of work in the last couple of years, some landmark papers in Nature in the last year looking at genetic determinants of the same protein panel we looked at in the UK Biobank through protein quantitative trait loci and things like that. There's a little bit less on trying to map exactly the environmental influence on expression of a lot or abundance of a lot of these proteins. Some people in our lab and other labs I know are working on things like that now. But in my mind, the reason why I think the proteins are a more sort of strategic, sort of omic layer to look at, if you will, is that they are at least the functional layer that is influencing the disease process or the frailty process or anything like that. And that's the really important part. And that's why I think that developing this kind of signature of aging from proteins tends to work so well as a morbidity predictor or a mortality predictor. You don't see that same phenomenon when you look at DNA methylation, for example, or transcriptomics. There's a lot of interesting research out there that shows that even especially for aging-related proteins or genes, there's this really strong decoupling between gene expression and protein expression. And if DNA methylation is going to matter at all, ultimately, it's got to influence gene expression, which I think explains why a lot of the early epigenetic clocks don't associate very strongly with mortality and morbidity if you just use methylation to predict age. So all the newest generations of methylation clocks will do different things instead. Like they'll train directly on mortality or morbidity or blood biochemistry or things like that as a way to get around that. But the proteins, on the other hand, seem to carry all that information directly. And, especially, like I said, some of the most important proteins in our model are extracellular matrix proteins. These are proteins with an incredibly long half-life, which means that after the gene is expressed and there's a protein, it's undergoing tons of post-translational modifications because it's just sticking around for a really long time.
Right.
And I think this is one of the reasons why they're so predictive for age. It's because they stick around so long. You have these alterations that accumulate with age and with time that are very predictive and very telling of what age you might be at. But that means, again, that they're going to be very divorced from the sort of upstream gene expression or possibly DNA methylation signals as well.
Yeah, it's fascinating. And I want to go on a little bit of a genetics tangent here just because I'm curious, but then we'll come back to this. There were many famous or infamous genetic studies that tried to look at super centenarians, very old yet healthy people, to try to find that fountain of youth gene or whatever. And none of them really ever found, at least to my knowledge, any smoking gun of this is why people in this particular area of the world live to be 110 and have no problems. Yeah. I'm wondering if why you think that kind of route of inquiry didn't really lead to much, or is there something there, but it's a scale problem or it's a phenotyping problem? Because it sounds like what you've picked up here is a really robust way to pick out, on a protein level, signals that drive pretty significant differences in biological age versus calendar age. But it seems like those are complex and driven by the environment rather than saying, okay, we found a subset of people that have a particular protective genotype and they've inherited this lucky fountain of youth. It's really something a little bit more complicated than that.
I think it's a really interesting question. I think it's a mixture of a lot of the things that you suggested at. I think there's definitely a phenotyping problem. I think there's not a lot of great supercentenarian data sets out there. There's an interesting preprint that's been floating around on bioRxiv for a few years now showing that among sort of supercentenarian data, a lot of supercentenarians in sort of demographic records are probably some kind of like pension fraud.
A clerical error, right?
Clerical errors or things like this. That was a paper that came out from Oxford. It's a pretty interesting paper. So there's definitely a phenotyping problem, and whether you're using those kind of records or not, it's difficult to get data sets with enough supercentenarian cases. There are some genome-wide association studies more broadly of lifespan and things like that. If you look at kind of the reviews and maybe the meta-analyses, there only tend to be 2 genes that I think pop up in any reproducible way, which are ApoE and FOXO3. The rest just don't seem to replicate across lots of studies. Interestingly, in our study, we have proteins for ApoE and FOXO3. Neither of those are selected by our model. Although ApoE is obviously a neurodegeneration influencing protein, we have plenty of other neurodegenerative proteins in our model, just ApoE is not. So there's some kind of disconnect between the genetics of this and the sort of downstream proteomics, at least. But at the end of the day, lifespan and aging are not phenotypes that I think are heavily genetically determined. The estimates vary across studies, but I think the best estimates I've seen put the heritability of lifespan somewhere between like 12 and 20%, something like that. We recently ran a genome-wide association study of our proteomic aging signature in the UK Biobank. We find the SNP heritability to be about 12%, so like right in there. There's other papers. There was a paper that came out in Science a few years ago on sort of millions of family trees, and they also found the sort of, at least the family tree heritability to be also around this 12%. Different types of estimates of heritability, but nonetheless. So all to say that ultimately, I think we're discussing a phenomenon biologically that is not very genetically determined, which is good because that means that, it's not really written in our genes, so to speak, how long we're going to live. There's a lot we can do to influence it, but that also means there's a lot that can be out of our control that can influence what trajectory we're on as well.
How do we square that fact with the fact that so many of these diseases that we're talking about do have a high heritability? I'm just thinking if lack of healthy aging is avoiding cancer, Alzheimer's, heart disease, and all these kinds of things, all of which have pretty high heritabilities. How do you think we end up with then the kind of composite phenotype being so low?
Well, I suppose it depends on what you would say qualifies as high. So another study that I was doing, this was more when I was doing my PhD. We have another paper that's up on meta-archive now, where within, again, the UK Biobank, we were looking at the relative proportion of variants explained for most of these major chronic diseases from genetics or really polygenic risk versus the exposome, which is just a word for all of the sort of contribution of all of your different environmental influences across life. And there we found pretty starkly, with some exceptions, that for most of these major chronic diseases, if you're putting sort of age, sex, polygenic risk plus the exposome in a model together, the polygenic risk is going to be explaining a minority of the proportion of the variants compared to just age, sex, and the environment. And we found that for premature mortality. We found that for a lot of major chronic diseases as well. There were some exceptions where the genetics trumps the environment. Things like Alzheimer's and all-cause dementia were two that stand out, like you mentioned. But I don't know. I see from at least some of the data we look at that when you're looking at the relative contribution of genetics versus environment for a lot of these kinds of phenotypes, there is for sure a reproducible genetic component to them, but it might not explain as much of the variance in these phenotypes as ultimately the environmental influence does.
So I was interested also in how you broke the score down. So you've got you've got 200 proteins and then even down to 20 proteins that you can get really great accuracy at predicting aging. But as you spoke about at the beginning, there's lots of different kind of branches of the tree. If you think about, we're heading down a healthy life, but then we take a detour off one branch or another. Or were you able to deconvolute the score in some way to say proteomic signatures that look like this actually tend to result in dementia more frequently down the line? Proteomic signatures like this tend to point towards cancers? Or is it this is purely an aging, and then beyond that point, there's lots of things can go wrong once you've sort of veered off the path, so to speak?
That's a really interesting question. We didn't do it in this paper. However, you could do it. The nice thing about the state of where we are now with these different models, at least machine learning models, is they're sort of moving away from being black boxes, and now we have sort of explainable AI tools for them. So we use in our paper something called SHAP values, which is just kind of a measure of each feature in the model's sort of contribution towards predicting the outcome. In this case, what's the contribution? And you get a quantitative score for each one of each protein in predicting age. But you get those values per individual person. So you can kind of explode these models and then look at, for every individual sample or person in your model, it was this protein that was contributing most strongly versus this person, it was that protein. The nice thing about a lot of the proteins, at least in this panel that we used, is that a lot of them have known connections to different diseases and physiological processes. So at least in terms of mining the literature, you could start to create maps like that. Although, like I said, we didn't do it, but it would be a really interesting idea. But you could probably also do a data-driven approach as well, which is maybe what you were hinting at as well, which is, kind of create different profiles in terms of which protein's working most strongly for different people and see if those match up to different disease profiles. It's a really cool suggestion. We'll probably try it now that you suggested it, but no, we haven't done it, so I couldn't tell you.
No, cool. That's great. I'm interested to see, like most ideas, it will probably require a lot of iteration before it goes anywhere. But where I was going with it was thinking about, you mentioned at the start of the call how this could lead towards, drug discovery, therapeutics type of applications. And I'm wondering whether those applications would target aging more broadly to say, we've learned from this what hallmarks of aging are, and then we can reverse engineer that to go after whatever the root cause of this is, or whether it's more likely to be we've reverse engineered this along a couple of the critical paths, whether it's neurodegeneration or cardiovascular disease. More of the, I guess, traditional, most drug developers would think about it that way, whereas a lot of the Silicon Valley people want to just go straight at the core, right? Which you mentioned isn't necessarily the focus of this. But I'm curious how you think about how this parlays into therapeutic development.
Well, in terms of therapeutic development, there is what would be ideal, and there's what's feasible or practical within the sort of regulatory landscape within the US at least. Aging and lifespan are not accepted indications.
Clinical endpoints, right? Yeah.
Right. So there's been a huge swell in the number of aging, longevity-focused biotech, even pharma activity in the last 5 or 10 years. And I think the route that a lot of them are taking is more the second example you gave, which is they're trying to understand something about aging biology, but they're funneling it through a very specific indication. And they're trying to pick strategic indications where this is likely something very age-related, such that some biology we're going to tap into might be more common across lots of different conditions. And then maybe we can go down more and more indications in the future and repurpose this therapeutic for lots of other things. You see that kind of happening inadvertently with, semaglutide and the GLP-1 agonists and things like that. It just seems like every day there's like a new possible application for these. But it doesn't surprise me, given that they are working on sort of this kind of like metabolic sort of adiposity pathways that are really shared across a lot of things. And
right.
By the way, when we look at the genetics of this proteomic aging score, some of the largest shared heritability, genetic heritability you see is with other kinds of like, metabolic, cardiovascular, like fat mass phenotypes. So it doesn't surprise me that would have many far-reaching effects beyond. But whether we will get to a phase where ultimately people are developing therapeutics on more broadly just aging, I don't know. It depends on how the sort of regulatory landscape evolves around this. But I think the evidence supports the idea that like, if we could go straight after aging, it would probably be the most strategic way to spend your money. You can develop one thing, and if it's really targeting this sort of shared common biology, it's going to have these knock-on effects across all the related conditions.
Yeah.
So I think would be a great place to go. I don't know how quickly we're going to get there, but one of the things we really need in the field to be able to do that are good biomarkers for aging. That was one of the reasons why we wanted to create something like what we developed. But just from a practical point of view, it's not feasible financially or time-wise to do a randomized controlled trial of lifespan in humans, which is one of the reasons why you see so much activity in mice and other animals.
Yeah.
If you run a 60-year randomized controlled trial on some intervention for lifespan or longevity, and then it doesn't work, you just wasted 60 years. That's possibly even more than one person's whole career. So
yes,
there's I think a real need for biomarker development in this field that can work strongly with those endpoints, but where you can measure much more quickly.
Right. I mean, it seems like you have one because you could, with the 20 protein score, you could imagine using this as a much quicker readout to get a picture on the impact that something is having on accelerating or decelerating aging, right?
That's our hope. We're working on evaluating this now within the context of a few randomized controlled trials with different endpoints for weight loss and BMI changes and things like that. So I guess stay tuned. Hopefully we'll have some interesting information about whether this really is capturing that in this context of interventions. But at least from a population risk perspective, it seems to work very well. And, on a genetic level, the genetics of this proteomic aging score are very strongly genetically correlated anyway with, the largest GWAS of lifespan and longevity and things like that. So we seem to be in the right direction, let's say, but there's some more work to figure it out. But this is there's a lot of activity sort of on this field in general in terms of trying to come up with good biomarkers of aging. I think right now it's like a kind of a holy grail a lot of people would really love to see, because it would totally change the way they could run trials and evaluate therapeutic candidates.
Absolutely. What do you think about causality here? And like maybe I can frame the question to how I'm thinking about it. So suppose we have someone whose the protein indicates that they're 60, but their calendar age is actually 50. So they've got a number of these 20 or 200 proteins that are at higher levels, let's say, than they should be. Would actually, let me flip it around. So we've got someone who's biologically younger than the calendar age says. If you were to spike their proteins into someone who's on the opposite, where they're biologically older, is it the level of the proteins themselves that you think could correct, and the level of the proteins is providing that protection or increasing risk, or are the proteins probably more likely just the canary in the coal mine, and what's actually happening under the hood is much more complicated, and spiking those proteins in, or literally bringing somebody into line with that score, isn't actually going to change their age in any way?
I think it's possibly a combination of both. There's probably some proteins in there that are just readouts of the disease process. However, that's one of the reasons why we wanted to evaluate the performance of this score among those who had no lifetime diagnosis of any major disease. And you still see just as strong associations with a number of different frailty, functional status, phenotypes, cognitive status, the different diseases. So, I think there is an indication that they are capturing something more upstream that's possibly influencing the morbidity process itself and not just a readout. Because if it was just a readout, you would see it among people who have disease already, and you wouldn't see it in those who are just healthy. And same thing, when we validate this in the FinGen cohort, where at least the way they've done the sampling for the proteomics so far, there's really no morbidity in that sample in terms of major diseases. There's like a less than a 1% mortality rate or something like that, maybe. So I think we have good indications that it's picking up some kind of causal disease process, but we can use genetics to tease that apart more. And that's one of the things we're doing now. And I think I think now that we have this sort of marriage of large scale human proteomics in, biobanks and data sets with large scale genomics and, health record data, things like that, you're probably going to see an explosion in our ability to use genetics as a tool for inferring causality for a lot of these things. So that's definitely an area where we're trying to move very, very rapidly.
I was curious, just as we close out here, how did you actually get interested in this in the first place? What brought you into aging and proteomics and set you on this path?
I have a very, very nonlinear path towards population science in general. I actually did my undergrad in philosophy and medical anthropology. And in the context of doing some of that research, which was more on kind of science and technology studies of how do scientists do their work and how do they know what they know, kind of like a study of knowledge kind of thing.
Yeah.
At some point, I realized I was actually more interested in maybe doing the science than like studying the people who do the science themselves. And I had the chance in those contexts to get my idea to sort of do my own hands-on work in some of these labs and things like that. And I really enjoyed it. But so eventually, for my PhD onwards, I moved into sort of population health, population science. But two of the things that really stuck with me from the anthropology were one, this kind of like holistic view that they really tried to imprint on at least, young students, that like, let's take this sort of bird's eye holistic view of how all the pieces fit together. And also just this ongoing interest in how do things in our environment hit our biology, so to speak. So I've always been interested in that, which is one of the things that has made me very interested in proteomics, like I sort of alluded to before, as it's this really good medium for being able to study the sort of like convergence of how are our genetics driving health and disease, and then how is the environment influencing those, which proteins are just good sensors of the environment, but they're not part of the causal disease process, and which of them are, and kind of like a mediator in that chain. So, and like I said, I spent a lot of my PhD focusing on trying to find the combinations of these, and try to work on statistical model development for bringing in environmental data and genetic data together. So that's kind of the legacy. But sometime in my PhD, I just started reading books about aging, and it just struck me that, like, I don't know, maybe I was thinking about it differently, but it just struck me that if there really is this common biology across all these common diseases, this must be the most strategic way we could invest our resources to try to curtail multi-morbidity rates, premature mortality, all these things which are major public health goals. So once I started thinking about it that way, it was hard to go back to studying just one disease or the other.
Yes, that's right. I think, and you're not the first of our guests that has a humanities history or literature type of background. And I don't think you are the last, because it's something about the way that it causes you to think about the world a little bit differently that allows you to come into science and just bring a fresh perspective, which is what it's all about.
I agree.
Just to close out here, what's next for you? What are you most excited about working on in the not too distant future? And how can people find you if they want to work with you, collaborate, all those sorts of things?
Well, I have a website. I think it's just austinargentieri.com, something like that. Put me in Google, you'll find me. Feel free to reach out. My email is there. I'm on X, formerly Twitter. You can find me there as well, or LinkedIn. But email is great. We're working on a lot of stuff now. Again, like I said, we're really trying to look towards the future in trying to drive this kind of new generation of leveraging all of the great, enormous, public health and biobank data resources across the world to study aging. I mean, I think in the population genomics category, or field, we've been on this for a long time, right? I think it's a given that people would work with the UK Biobank or FinGen or any of these major biobanks, but the aging field hasn't quite got that message as much. So we're trying to really work towards leveraging bring together a lot of different cohorts and other types of multi-omics data as well. One of the most common questions we get all the time on our work is, how does your protein clock compare to all of the epigenetic clocks that are out there? And unfortunately, the UK Biobank doesn't have any epigenetic information. So our next step is definitely trying to do more multi-omic comparison, tease that apart in a, like a large human population context. So
amazing.
Lots of interesting insights from that. We're going deep now on the genetics of this proteomic age signature that I mentioned, that we're, trying to pull out what insights we can there in terms of how there's common biology and pathways across lots of different conditions. There's lots of interesting stuff we're finding on there so far. So I would say stay tuned. There'll be some stuff out on that in the near future.
Very cool. Congratulations on the great work. And thank you so much for sharing it with us.
Thanks so much for having me. It was a great talk.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.